Welcome to the second session. My name is Sunil Gandhi, and in this session entitled Emerging Resources for Team Science, we're going to go through a mix of rapid-fire talks to introduce you to some new and emerging resources that are available here on campus to support new team science initiatives. I'm really excited about what we're going to be discussing today. In the interest of time, we're going to, I'm sure that each of these talks are going to generate a lot of questions. We'll probably just go through, and perhaps there'll be time for one or two questions, but I think let's try to save them for the end. And if your burning question doesn't get answered, please be sure to seek out the speakers afterwards during the lunch and brainstorming session. I'm sure they'll be thrilled to answer your questions. So first up is Dr. Melinda Gormley from the Dean's Office in the School of Biological Sciences, who's going to be talking about dimensions as a team science development tool.
Well, while Sunil brings up the presentation, I do want to bring your attention to your programs. There is a page on dimensions in it in case you want to refer to it for additional information. And you have heard a little bit about dimensions from Mike Yasa earlier, just a few minutes ago. So one of the main goals for UCI Brain is team science and interdisciplinarity. We've heard a lot about this so far today. And so one of the things that UCI Brain also wants to do is invest in resources that are going to allow you to do team science. And Dimensions is a platform that really helps you to formulate teams, find collaborators, as well as find funding that you might not otherwise know about that can fund your projects. So this is an example. I'm using screenshots for this presentation. This is Dimensions. What you see here, Dimensions is a system in which it pools various information. It actually has hundreds of partners that are international. It presents information and pools information about publications, grants, patents, clinical trials, policy documents. And if you were to go to Dimensions and try to access it, the publications is free. Anything that you want to look up and different kinds of ways that you want to look at the publications, anyone can do that. However, there's the real exciting capabilities of Dimensions are behind a paywall. And that's looking at grants, looking at patents, clinical trials, policy documents, but really the analytical views, which is over there on the far side from me. And so one of the things that UCI Brain is going to give is access to Dimensions, to these other aspects of Dimensions. How? If you go to the UCI Brain website and you push on the resources tab at the top, the first thing underneath that is Dimensions, and it'll bring up this page. And as you'll see at the bottom of this screenshot, how to access Dimensions is there. If you ask UCI Brain for access, you will within one business day most likely have a response in order to help you with that, to get into there. Also, there are different levels of access, and one of the things that UCI Brain is going to do is if you do submit for a pilot award and you receive it, you're going to have those greater capabilities to help you with with the team science work that you're doing, and even some of the competitive proposals that are not funded will also be able to have access to UCI Brain's subscription into Dimensions. There are other resources, and we've heard a little bit about this today besides Dimensions, and before I go into more about Dimensions, I do just want to bring your attention to a couple of additional resources. UCI's Team Science Consulting Services, there's information through through ICTS. Also, another resource that is mentioned a lot by Dan Stokels, Judy Olson, Gary Olson, Maritza Salazar Campo, is this NCI, or National Cancer Institute, field guide on team science. And UCI Brain has hard copies of this document if you are interested. It's also available in PDF for download from the National Cancer Institute website. All right, so a little bit more about dimensions. My favorite part is grants, since I'm Research Development Officer and I help faculty in the School of Biological Sciences get funding. One of the things I want to know is who has funding, where is it coming from, how many different grants, what size, and that's something I can get in an instant through Dimensions. What makes it different than other sites? If I go to NIH Reporter, I'm only learning about NIH. If I go to NSF, I only learn about NSF. Here, it's pulling internationally, foundations, federal agencies, and so I'm getting a full picture of an individual's funding, an individual's publications, and just who's working on what and how they're doing that. So the analytical view, before I go into the analytical view, if you were to push on, say, Alzheimer's Disease Research Center at University of California, it would then take you to more information about that, as well as it would take you to NIH. You could go out to NIH Reporter from Dimensions. I have limited my search to UCI, neurosciences, and ongoing funding, so that means funding that has not been completed yet. When I open in analytical views, I push on research categories, and I choose bar chart. I get information about how many millions of dollars UC Irvine has in terms of neurosciences funding. They've broken it down in dimensions into these different categories. It might be a little confusing what actually falls under these categories. So if you look over there where it says chart and table and click on the table view, you can actually drill down into what kinds of subcategories are in each of these larger headings in the bold. One of the other things that it has, and you can see this over on the side closest to me, there are different tabs in terms of getting an overview of funding. You can actually look at which researchers have how much money. If you were doing this and it was not limited to UCI, it would just tell you which research organizations are coming to the top. And you can also compare two institutions. So you could compare UC Irvine to UC San Diego, UCLA, or another institution to see how they both are operating on a particular topic or just in general. So one of the other analytical views here, what I did was to go into where it says overview. And these are two different shots, one of the chart and one of the table. Because I'm looking at ongoing grants, this is showing me all grants that are current today, shows me when they started. So of course you don't have many grants started in 2010 or 11 or 12 or 13 that are still ongoing today, because something like an R21 would have already completed if it started back then. But it also gives me an understanding of how many grants have started in the last couple of years, how many are of active grants in neurosciences at UCI there are currently. And then it has those different comparisons. If I were to take off ongoing, this would look more like a straight line, and it would show all of the funding that is completed as well as is ongoing in terms of neurosciences at UCI. So one last thing about UCI Brain and Dimensions, that you can learn more. One way to learn more is to come see me. I'll be sitting at the table outside. And as Mike said, I will look you up if you like. I looked myself up the other day and learned something about myself that I did not know. It was accurate. I mean, it was actually me. I just didn't know it. You can request access. Again, you can do that through the UCI Brain website, through the Resources tab. And then there will be a tutorial in which the Dimensions, people from Dimensions will come here and give a training. And I am not paid for by Dimensions, but I'm a huge fan of Dimensions. So you can also, again, come see me anytime today. For most of the rest of the day, I will be sitting outside at the table with this up on my computer. I have full access, so you can see all the things that are behind the paywall and look yourself up, check something else out that you're interested in, and just come and ask questions. Thank you.
Okay, so next up we have Mathew Blurton-Jones. Okay, take it away, Matt.
Thank you very much, Sunil, and thanks, Mike, for the invitation to come tell you guys a little bit about this. Let's see, how do we get this up here? There we go. Okay, so some of you may have heard of this, but if not, this is part of the reason I'm here, is to tell you a little more about a new core that's been established at UCI just a little under a year ago, actually. And this is through the Stem Cell Research Center. So this was encouraged by Eileen Anderson when she took over the directorship of the center. You can see is one of the websites where you can look at resources and find our CRISPR core resources link there. So what do? Well, we can engineer induced pluripotent stem cells to modify a gene. So for instance, we can knock out a gene, we can knock in a mutation, we can make reporter lines. And we started with iPS cells because that was sort of our background, but we've also continued to start to do this with other cell types. So if you're not working with iPS cells but have other cell types that you're interested in, as long as they're proliferating, we should be able to help you with this. I don't want to get too technical here, but I just want to very briefly explain for those of you who aren't familiar with CRISPR what it involves. Essentially, the breakthrough with CRISPR was the ability to cut DNA on both strands in a very targeted fashion. And so you can use these guide RNAs to deliver this nuclease called Cas9. You can target it to a specific gene. That makes this double-stranded break, and then there's a couple ways that the cell will try to repair it. One is called non-homologous end joining, the other is called homologous recombination. And we can take advantage of these two repair mechanisms depending on what our interests are. So if we want to make a knockout, we can just let the cell try to fix it with non-homologous end joining, and mistakes are often made that lead to stop codons and therefore a knockout. Alternatively, if we want to fix a gene or change a single amino acid, for example, we can add a template which the cell will then use on the right side through homologous recombination. So with all these steps, we sort of induce a double-strand break and then allow the cells to repair, and we kind of help them along the way depending on what we're trying to achieve. The next step then is, of course, to screen and identify the actual clones at a clonal level, single-cell level, that we then expand to find cells that have been appropriately targeted. So there's, of course, more detailed steps, and we have on our website this link that shows us sort of the steps we take through. For example, in this case, for a point mutation introduction, we have conservatively said that this takes about 2 to 3 months. We're finding it's closer to 7 to 8 weeks, actually, so we're quite efficient at doing this. But it starts with a consultation, asking the researchers about the gene they're interested in. Then we'll design the guides and validate the guide's ability to cut. And then we can go sort of a full service run through. We can also do pieces and parts, depending on the researcher's interests. Now, CRISPR, some of you may know, is a fairly expensive proposition, especially when you're working with iPS cells. So we do have a price list on this, and this has been supported by the Stem Cell Center to keep our prices low. And sort of as proof of that, one of our closest competitions here is actually the Harvard Stem Cell Bank. And if we look at the pricing, they're actually charging about twice as much, even for their own internal users. So I think we're giving UCI a fairly good deal in terms of generating these cells. So how are we doing? Well, we started just back in January 2019, and we're already making quite a lot of progress. Up at the top, you see some completed projects that we've done for various researchers here at UCI. We've got several ongoing projects that are moving forward as well. But we're pretty sure we can handle more, so if you guys are interested, feel free to reach out to us. Now, being a researcher myself, of course, I feel uncomfortable not showing a little bit of data, so I do want to show one example of how CRISPR technology is being used at UCI. So just the last couple slides, I want to show you how some work being done, and in this case, this is in partnership with UCI's Alzheimer's Disease Research Center that helps us to provide samples from patients who have or don't have Alzheimer's disease. And we were interested in studying this gene called TREM2, which has been shown to be a risk factor for AD, so it increases risk of Alzheimer's about threefold. So we can start by trying to make a loss-of-function mutation, essentially a knockout, by using CRISPR, allowing non-homologous end joining. We can screen our clones, in this case looking at chromatograms. The top is showing the wild-type alleles. The bottom two is looking at the two different alleles to show that we have loss-of-function mutations, so we induce stop codons, premature stop codons in both alleles. And we've done this, in this case, with 3 different individual lines and validated at the protein level that, yes, we've made a knockout of this protein. So what can you do with it? Then you can start to do things like in vitro studies, in this case, taking microglia that have been generated from these iPS cells, feeding them with beta amyloid that we know is very important in Alzheimer's disease pathogenesis, and finding that this knockout mutation impairs their ability to phagocytose this amyloid. So that gives us some indication of how TREM2 might be influencing Alzheimer's disease. We can also actually move this forward to some in vivo studies. So, for instance, by transplanting some of these cells into immune-deficient mice, we can actually make a chimeric model to study human microglia, iPS-derived microglia in vivo. And when we do that, we see some very interesting features. So normally in Alzheimer's disease, microglia will migrate to the plaques and surround them. That's what you see up above with the wild-type cell. But if we have the knockout cell, and this is again from the exact same patient, just that knockout change, the microglia no longer pay attention to the plaques. They don't migrate to them. And this has a lot of effects on their gene expression. They seem to be locked in a more homeostatic state, so they're not responding to the pathology the way they should. So this is an example of using CRISPR, making an iPS line, starting to use this for both in vitro and in vivo studies to learn about that specific gene and how it might be influencing Alzheimer's disease. So with that, I just want to end with our email there. Feel free to reach out to the core members. John Paul and Lisa are really the ones running the show. They're amazing, incredibly efficient, incredibly bright, and we're happy to work with you guys. One important thing is we're also happy to provide letters of support for grant applications and even can provide some preliminary data if that's useful for feasibility. So thank you.
Okay, great. Thank you so much, Matt. Now I am going to do the perilous thing of introducing myself as the next speaker. You know, we were meant to have a squawking chicken here to keep people on track for time, but the chicken somehow did not show up today, which is fortunate for me because now I'm going to give a talk. No chicken for me. Okay, so in this talk, I wanted to introduce you to some new resources and technology that my laboratory has been developing in collaboration with Zeiss Microscopy and also a recently developed UCI startup company that I should disclose I'm the chief scientific officer for. That's Translucence Biosystems. And what we have done is to develop some new technology that's meant to change the way that we do brain histology, to bring us into a more modern, three-dimensional perspective. And I'm excited to share this with you and give you some examples of what we've been able to accomplish here in the optical biology core, which houses the system that I'm going to describe. And if you are interested, the OBC, the Optical Biology Core, has a table outside, so please do and go and seek out Adila Saeed to find out more. Okay, so the brain. We want to understand the brain and its mysteries, but unfortunately, in many ways, the brain is opaque. It's quite literally opaque as a piece of tissue, and to look at its cellular organization, for hundreds of years we've resorted to chopping up the brain into thin sections and laying it out on slides so that we can look into it with a microscope. But in the last 5 years or so, new technology has emerged that allows us to hold the brain together and keep it intact while making it optically transparent. And in this tube on the right is a whole mouse brain sitting there, but it's been made transparent by a recently developed clearing method. Once we've done that, now instead of chopping up the brain with a blade to look into it, we can instead use a sheet of laser light to rapidly illuminate the brain and fully visualize it. And so here's an example of a brain that we have cleared and imaged on a light sheet system that we have developed in collaboration with Zeiss Microscopy that is much faster than any of its predecessors. So here you can see this particular mouse is a Taiwan EGFPM mouse in which a smattering of neurons are labeled with a fluorescent protein. And here you'll see the structure. Here's the motor cortex and then hopefully we'll see the descending individual projections of the corticospinal tract. This video goes on forever. I will not show it to you, but it's amazing what we can see in the brain. This technology is very exciting. I should say that it's quite rapid. So this data was gathered in 20 minutes of imaging. So the technology is quite powerful and opens up new doors. But it's more than just for making cool movies, although I do find that to be an entertaining activity. What I wanted to tell you about today was a little bit of this technology and how UCI collaborators are taking advantage of this technology. I'll give you a few examples. And then I wanted to tell you about two stories that were very recently published. Matt introduced you to one of them, and then the other, a collaboration with Melissa Ledun, that I think illustrate the power for this technology. So the first thing is to show you something more about how we can use this technology. So what you see here is a mouse brain and spinal cord kept intact. You may also notice that we've left intact one of the nerves off of the spinal cord. That's that winding tissue in this middle panel. I don't know if we have a pointer. I blind myself. Okay. In this panel here, you can see the fluorescence image taken with the light sheet of the cerebellum and brainstem, and then the spinal cord with a number of the cervical ganglia left intact. Here's a zoom in that shows you where one of my students placed a viral injection in vivo to label some of these sensory neurons, which have a peripheral process that innervates, in this case, the forepaw, and then these central axons. My student had imaged, actually, in vivo, using multiphoton imaging, a segment of this, and then he was able to reconstruct the entire neuron using brain clearing from that very same sample. So this shows you that there's tremendous power in this technology to see in 3D structures that previously with thin section histology we would have had a great deal of challenge to look at. And this technology is not only cool for getting this sort of mesoscopic view, but also for allowing us to, keeping this tissue all together, zoom in and find all kinds of microscopic detail. So here is a zoom in on the light sheet data from one of these cervical ganglia that was labeled. And what we're going to do is here digitally dissect and flay open that structure and go in and see here actually the meeting point between the central and peripheral processes of one axon, and then we can look at the same movie now, the same data sample on the confocal system that's sitting next to it in the optical biology core. This very same T-junction here, now with confocal resolution, and we can molecularly interrogate that using a stain for a particular ion channel that you'll see will turn on in a second, that together tells us that this particular T-junction, buried deep in this structure, is a nociceptive fiber. It's a pain fiber. And this kind of traversing between the micro to the meso scale is something that we now can do, and its potential is, I think, quite vast. So here's another example of work that Ozzie Stewart has done together with us, in which he's labeled the motor system by making a retroviral AAV injection into lumbar cord here that illuminates the entire motor system. If you look at the brain, you can see all these lovely structures of cells. Sorry, the movies are looking a little funny. But what you would have seen in that movie, there I go back, is that, let's see if we start again, that in addition to the motor systems, there's this hunk of label right here. And that hunk of label turns out to be the pineal gland, which is, I guess, the seat of this mouse's soul. And for whatever reason, it's taking up this retroviral AAV injection. How it does it, Ozzie is he here? I don't know, to speculate on why. It's kind of fascinating because this has incredible implications. just alone for AAV viral therapy, that into the nervous system, if it turns out that there are certain structures that are susceptible to viral infection that you never would have guessed. Anyway, just some of the kinds of things you see when that you find when you're not looking for them, when you start looking at the brain in 3D. Okay, so next I want to tell you about a collaboration with Melissa Ledon's lab, in which we wanted to understand how a parasite that has a major impact on global health, toxoplasma, also a model for malaria, how this parasite manages to infect the brain of mice and turn them into cat lovers. So it turns out that when mice are infected with toxoplasma, they lose their fear of cats, among other behavioral abnormalities, which is quite good, not for the mice, but for the toxoplasma, because the parasite requires the gut of the cat in order to reproduce. So this is a very elaborate matchmaking service that the mice provide. And we wanted to understand how these cells were altering mouse behavior, and there really wasn't a good neurobiological explanation out there. And Melissa in my lab discovered that, that what happens when the animals get infected with toxoplasma is that there's this sudden infiltration, there's a stickiness of monocytes here all of a sudden in cerebral blood vessels that then results in the infiltration of monocytes into the brain parenchyma. And this form of invading inflammation was something that we wanted to map out. Maybe it was going to certain regions. So we used this technology of brain clearing and light sheet imaging. And this is an example of the workflow. These are the monocytes in the brain. We can use the autofluorescence channel in order to align each and every brain to the Allen reference atlas so that we can precisely quantify exactly where these cells landed and automatically across 700 plus brain areas that are in the reference atlas. And this is how we look at the data. So here's just an example slice of the brain and a zoom in to show you how high content this imaging is. Each of these little chevrons points to a monocyte. We can segment out using machine learning tools all of these hundreds of thousands of monocytes in the brain in 3D. And then quantify precisely where they're located by aligning to the reference atlas. And this is what the data looks like. So each column here is the density for a different brain that we image. We image 5 brains in a morning. And it shows you each row here shows you a different brain area. So sorry, you can't read it, but that's because there are a lot of brain areas. There's brain areas in here I'd never heard of before, actually. It's kind of cool to go through this list. And so this data shows us that there is kind of a sporadic, irregular pattern of expression. But maybe for some of you in the audience, it reminds you of something. Maybe it reminds you of sequencing data. Well, we thought maybe we could analyze the patterns of monocyte infiltration by using some of the statistical framework that's used in sequencing data. So we use that to discover that the infiltration of monocytes seems to go into really only one brain area consistently, a brain area called the olfactory tubercle, a place I had never heard of. But now I know that this amazing brain area sits between the olfactory cortex and the ventral striatum and plays this enigmatic role in regulating behavioral flexibility, motivated behaviors, and perhaps you could imagine focal inflammation into this area perturbs the animal's behavior so they stop losing their fear of natural predators like cats. So this is pretty amazing, and this is something that generates a new hypothesis, and that's exactly what you hope for from an unbiased search technique, and something that I think could be quite promising and interesting for all of you in your own work. So just quickly touch on how we've used this technology for looking at human microglia that Matt introduced together this project with Matt Blurton-Jones and Rob Spitali, we were looking at at a chimeric preparation in which human microglia were delivered into the brains of mice so that we could study these human microglia in vivo, but without opening up any human brains. And there's much that we can discover here, but I just wanted to show you how we use this technique to map out where all those microglia go. So here is just the raw data off of the light sheet imaging showing you all of the beautiful detail that we can visualize, again, very quickly in the mouse brain of these human transplanted microglia. We've also been collaborating with others, including Mehrdad Shamloo, to look at patterns of neuroinflammation across the brain, in this case, induced by a systemic neuroinflammatory agent. And what you can see is that we can find very specific patterns of inflammation in certain regions, and it's quite exciting for us. Well, beyond localization, we can also use these machine learning tools to pull out the structure of each of these cells and use that to assess something about their state of activation. So the overall view is, let's say you have come up with some brilliant new compound that's the Tylenol for the brain, and you want to test this out. How are we going to show that it has anti-inflammatory effects? Well, what you can do is use this new imaging system that we've created to map out patterns of inflammation, say, in the form of microglia across the brain, and then see across these hundreds of brain areas how your substance has reduced the inflammation. This is just one of many possible applications. So just to wrap up, I wanted to mention that although right now we don't have a resource per se other than collaboration to support getting people involved, the Optical Biology Core, of course, has resources and is ready and willing to support people in learning how to use this new technology. But I wanted to mention that in June 2020, together with Zeiss Microscopy and my partners Pavan Kadandale and Andy Obenaus, who you're going to be hearing from next, we're creating a course for the international community so that people can come and learn how to perform brain-clearing light sheet imaging and how to connect that on a different scale with high-resolution MRI. And so with that, let me stop and stop myself, which is my job right there. Thank you. Okay, so next up is Andy Obenaus from Pediatrics, and he is going to be speaking to us about the Preclinical and Translational Imaging Center.
Thank you, Sunil. And now that Sunil took my 10 minutes, which is a hazard when the squawking chicken doesn't show up. So, but anyways, it's very fantastic stuff. But the one thing that I'm going to be here to tell you a little bit about is what if you don't have to clear your brain in order to see in the brain. And that's what we've got here today is UCI has invested in a preclinical and translational imaging center. And that's here to help researchers, particularly my own interest as well, brain researchers to investigate using MRI non-invasively, both in vivo and ex vivo, to sort of parse brain function. And first of all, it would be remiss if I did not thank the partners who have, over the last several years, contributed extensively to this effort. As you may know, MRIs are not cheap. And there's a lot of infrastructure that goes with that. And so I have to thank everybody across campus. And this is truly a cross-campus facility that can be used by everybody on campus. So again, many thanks to those partners and colleagues who have helped make this work. So what does this look like? So the instrument in the Preclinical and Translational Imaging Center, the first one, I hope, of many, is a 9.4 Tesla MRI. This is about 3 times stronger than the nice fancy fiber. And of course, with that, you get increased resolution and some other advantages. So it's a fairly large bore instrument that will be able to allow imaging for a variety of rodents, small dogs, cats, the cat that was going to eat the cat, or the mouse there, we could image that, for example. And of course, we can do lots of in vivo imaging, which is its power, but we can also do things like MR and MR microscopy. So if we image for very long time periods, we can get down into the 75 to 50 micron range, depending on how much time and effort we want to spend on that. The other thing is that, of course, we have a variety of components that help facilitate this research. So we have what are called radiofrequency coils. These can be designed or purchased based on your research needs. We have about half a dozen different kinds of these for various researchers. So we have ones for mouse, rat, and we even have larger ones for larger mammals. We also have microgradient inserts that allow us to get faster and better imaging sequences, and we can insert these as well as with these in combination and really try to improve resolution and quantitation. The next few slides, I just want to show you some of the capabilities of what this instrument can do. So for example, many of the things that we do are related to relaxation in the brain. So this is a rat brain here, of course, and it's T2 relaxation. You can do T1. One of the things that we'll be able to do, and I'm looking forward to this, is basically arterial spin labeling. We can look at how blood flow changes in the brain or throughout the brain. We can also do MR spectroscopy. So if there's a particular metabolite, for example, like glutamate or lactate or some other metabolic component that you're interested, using spectroscopy, we can actually go and parse this. And the nice thing about a high field, the higher field you get, the more clean these line widths get, and we can actually differentiate many of these metabolites. We can also do continuous ASL, just another type of arterial spin labeling. We can do lots of short TE kinds of imaging for anatomical structures. We can do angiography. With the right sequence, again, we can map out, and along with either contrast agents or without contrast agent, you can actually map out the cerebral vasculature in the brains of rodents. We'll be able to do what's called functional magnetic resonance imaging, which allows you to look at connectivity or regions of the brain that might be activated during a particular sequence. Let's say, pain or other kinds of things. We can use this to map this and then look at how different brain regions are connected together over time. And then we can also do fat and water imaging if you want to look at regions outside of the brain, for example, in the torso. And then one of my own favorites, of course, is diffusion tensor, which allows you to map structural connectivity between brain regions. And we've been using that with great success, and I'll show you some of this in a moment. And then finally, of course, there are cardiology applications. This is very daunting, of course, because the heart in a rodent is very, very, very fast. So this is not my favorite imaging modality, but if there's researchers on campus, we'll definitely work with you to try to do these kinds of imaging modalities. So I just want to give you, since everybody took the liberty of showing a little bit of science, I'm going to show you just a very tiny bit here, just some of our own work that we've done in collaboration with folks on campus. This is work with regards to febrile seizures with Dr. Tali Baram that we've done. And over the years, we've essentially done T2 relaxation imaging to identify a imaging abnormality that's located in the basolateral amygdala right here. If you see, this is a lot brighter here than it is over here. And the nice thing with T2 relaxation imaging, we've published a number of papers now where we're able to actually very cleverly identify brain regions that are highly susceptible for seizures in those animals that go on to have subsequent epilepsy compared to those that do not. So the nice thing, this kind of groundwork led to a funding of an R21 in children that we're now pursuing to actually pursue this signature in children. So that's a good example of translation. One of the other things that you can do is susceptibility-weighted imaging. We heard a little bit about iPS cells. If you were to incubate those with a contrast agent such as gadolinium or iron oxide or other kinds of things, you can inject these and then track them over time as they go and, let's say, invade an area of ischemia, as we've done. So we published a number of years ago on how to do this, and you can see, actually, you can identify this loss of signal where these iron oxide cells have been invaded. You can also do the same thing in looking at beta amyloid deposition in the brain. So amyloid oftentimes, in many cases, is associated with iron depositions, and here's the MRI. And this is a Prussian blue stain from the same animal, and you can see these Prussian blue stains. While we don't have the same resolution as microscopy, we can get certainly a very good handle on these components. And then also diffusion tensors. So this is a model of concussion. And you can see here, these are the different diffusion tensor metrics. We can get the T2, the fractional anisotropy, the apparent diffusion coefficient, the axial diffusivity, and the radial diffusivity. And if you look at first blush, many of these are not different. But with quantitation using atlas-based metrics, we can actually identify changes within the corpus callosum or the anterior commissure here and the corpus callosum using diffusion tensor in these sequential time coordinates. The other thing with diffusion tensor is, as you saw, we don't use the Allen Brain Atlas because the resolution's too high. So we use the different kinds of atlas that are basically set up for MRI. We can parse out cortical, limbic. We have an atlas that basically has about 80 different regions. And you can then kind of get these sort of heat maps after concussion type injuries, and you can look at basically how those changes over time occur between brain regions. We can also do what's called tract-based spatial statistics in these animals as well to identify white matter changes, again. And then this was in MS model. And then here is essentially treatment modalities where we have a fluid percussion injury for TBI, and this is tractography. So after giving it a treatment with these alavanoids, what we found is that the white matter tracts were much more preserved in these animals than those that were not treated. So just in summary, again, we have MRI that allows you to do T1, T2 kinds of related relaxation, angiography, diffusion tensor, functional MRI, perfusion-weighted spectroscopy, and there's a whole host of many things. And one of the things that I think is a real strength of in vivo or even ex vivo MRI is that we have this sort of menu of modalities that you can go and then look for brain function and activity. And so I will close with just letting you know the instrument has been installed. We are ramping this instrument up over the next month or so, and we hope to be open for business starting January, February for routine imaging. So if you have any questions, we also have a table with a brochure out there. There's a slideshow going on for some questions. If you have any questions, let us know. If you're interested in MRI, you've never done it before, and want to know how to start, give us an email or call, and we'll get you on the road to doing in vivo imaging. Thank you.
Okay, thanks so much, Andy. Okay, next up we have Dr. Kristina Uban, who's from Public Health, and we'll be talking about interdisciplinary salivary bioscience.
Thank you so much. So I know we're not supposed to harp on you, Mike, but you got to add public health and ISBR to your comprehensive list. So my name's Kristina Uban. I'm here representing a fantastic team of faculty here at UCI, and we're all part of the interdisciplinary efforts to enhance salivary bioscience research. Our director is Doug Granger. And we have several core faculty here that we all pretty much live in social ecology and in public health. And Doug Granger, Jenna Reese, and Elizabeth Thomas, I'm going to share some of their ongoing projects today, and all of them are away at conferences and are very disappointed to not be here. We have Kate Kuhlman in our audience, Alison Zalta's coming by, Ilona's coming by, and Michael Hoyt is also in public health with me. So the same excitement that we all have here for neuroscience, and I'm a developmental neuroscientist, I share that with all of you. That's the excitement we have for SPIT over at the Institute for Interdisciplinary Salivary Bioscience Research. So the same aspirations that you guys have and the moonshot ideas that we all have and that we're feeling supported here today to actually maybe act on them, that's what ISBR, that's what we hope to do for saliva and moving that forward. We have a fantastic team of staff. If you want to contact staff, you can contact any of us faculty members. All of our information's up on the ISBR website. You can contact any of us. We're all happy to help. Caitlin Smith is the manager. She's on maternity leave, so Hillary's filling in, and Tatum are the ones who really do a lot of the business side for ISBR and do all of the assays. They're fantastic. So we're over in Social Ecology 1 here at UCI. We're a huge lab, and we're a shared lab. So I started my faculty position here as a junior faculty member a year and a half ago, and I walked in to having a wet lab that I can call my own that has fancy tools to do really fancy things that I would never even have gotten access to those, even as a senior faculty member. So that's what a community lab allows for all of us. So it really facilitates our careers, but it also facilitates all these collaborations. So if you look and see your research interests and if they align with any of ours and you want to kind of dive into salivary biosciences, or you have in the past and you just want more support in doing it, this is, a shared lab that we all have. We offer a spit camp. It's a 3-day, all-day intensive training, mostly attended by faculty who want to really accelerate their ability to use salivary biosciences. Also, postdoctoral fellows will attend, and you can register up on the website. I have the website address there. I also teach a course for graduate students. It's open to anyone in any department. You register through public health, and it's in the wet lab. We teach it in the wet lab, and even if you don't have wet lab skills, we'll make sure that you are up and running independently by the end of the quarter. That's in the spring. Okay, so I went from animal models, and a lot of things I wanted to look at had to be done in trunk blood, had to look at the actual brain tissue, post-mortem, and I used to think of that really as the gold standard for really understanding what was going on in the brain. And then when I wanted to look at maybe, the peripheral measures of the neuroendocrine functioning, I would then collect blood from my animals, and I could do multiple assessments using the blood collection itself as the stressor to induce it, and then follow up after a certain time period. And all of these are great options. Now that I'm working with kids using multimodal neuroimaging, I've really come to have a better appreciation that the body supports the brain. And these peripheral markers that we're measuring in saliva aren't just kind of really downstream effects indicative of what's happening centrally. They stand alone on their own, because our brain is only as good as the body that can bring oxygen and nutrients to it. So saliva has become kind of the main focus that I've been using to look at biomarkers and connect it to what I see in neuroimaging data. The other reasons that we're really excited and geek out really hard over saliva at ISBR is that it's non-invasive. So people are spitting in tubes, and you don't need a phlebotomist, you don't need any specific specialty training to do this with people, and you can actually have them do it themselves or teach their parent to help them do it. That means you don't have to do it in the lab. My husband's a comedian, so I actually had a grad student during the wet lab course last spring come to our comedy show, and we had volunteers self-sample, and what does watching comedy do to you, right? So we did pre-post, did the comedians and the audience members as a fun learning opportunity, and so you can bring it to all these hard-to-reach populations. There's a physician in the audience that has an elderly or hard-to-reach population. You can look at all these different neuronal markers or indicators of health to people who can't necessarily come into the lab very easily. There's a fantastic poster out there on sleep circadian patterns. We can do all of that. You can send someone home. They don't have to come into a sleep lab. You can have less participants, less animals. You can sample animals. I can talk more about that too, or find me. I'll be here all day. But you can do more with less participants because you have that within subject repeated measures. And so statistically, you just have a lot more power to look at these things. All right. Here's a list, and is not comprehensive. One thing that we really like at ISBR is that all of our faculty members have unique interests, and Elizabeth Thomas, I'll share some of the stuff she's working on, but she works a lot on the development side. So if someone's doing it in brain tissue or blood, she will figure out how to do it in saliva. So here, I look at a lot of the hormones. So I do a lot of hormones. This is all up on the ISBR website too. But people in the ISBR faculty crew will look at therapeutic drugs. So you can have someone self-sample at home and see if the drugs are working. So you can look at all these different markers of disease progression. Drugs of abuse, we can measure cotinine, THC, any drug of abuse you're interested in. And in fact, other studies are doing that. So you can look at differences in metabolism. At a very fine graded timeline within an individual in terms of how they metabolize these. Environmental chemicals, We're all, I'm sure, equally just disturbed by the amount of chemicals and toxins that we absorb every single day. And of course, little kids are so small, so they absorb the same amount, but their bodies are tiny, so they can't, the same amount does a lot more harm in them. So we can take saliva from any age, from newborn all the way up to elderly, and we can assess all these different kind of environmental chemicals, from endocrine disruptors to a whole panel of metals. So Dr. Elizabeth Thomas has really been innovative for adapting blood and analytical machinery to do it in saliva, and the same volume of saliva, you can get a whole panel of metals, like lead. Excuse me, I'm getting over a cold. So, of course, other things that might be of interest to people, and I just want to say disease progression. So any markers of disease, you can look at DNA, epigenetics, and then I'm going to talk a little bit more in the other slides about the neuronal markers and uric acid. Another service, if you have saliva or you're an animal researcher and you know that human researcher who has a whole study going on, and if they could just collect saliva for you, it's very cheap, it's easy to implement, but you don't have money to analyze it yet, we can store it for you. We have a huge freezer farm. Okay, so I'm going to just, I don't have enough time to highlight research from everyone in our group, so I'm just going to highlight two. One is Elizabeth Thomas, who's away internationally at a conference. She does a lot of work looking at neurodegenerative disease proteins in saliva. But you don't just have to look at someone in neurodevelopmental disease. You can look at neuronal markers that are common to all of us. And you're looking at this in saliva. So why would these neuronal markers be in saliva? Well, if someone has disease, there's obvious blood-brain barrier breakdown, and so things that should stay central go peripheral. That makes sense. But she also sees it after just common concussions. You can see it in the average individual. So this really is such a moonshot idea for us that maybe we could combine for a moonshot idea with you guys, and we can land some undiscovered planet far away. But why is the blood-brain? Sorry, that was so cheesy, you guys. I was like, do I share that? I don't know. Why is the blood-brain barrier opening and closing? Does it do this on a circadian pattern? Does it do it once and then it just stays? Why are these in your periphery? So I work with kids with fetal alcohol spectrum disorder who do have organic brain damage, so I'm exploring these kind of neuronal markers in their saliva. Elizabeth Thomas, looks at individuals with Huntington's disease, brain injury and no injury, and looks at the ranges of these neuronal markers and sees how many she can detect disease states in. So she's really pushing the field. And she also is working with a lot of different samples, disease patients, Division I water polo players, Alzheimer's disease, and military personnel with the Navy. Now, Jenna Reese is working and done a lot to push forward how we understand uric acid. But uric acid is interesting because it's involved in all of these physiological systems that are so important for health. Everything from antioxidant activity, inflammation, to blood pressure. So because it's involved in all of these, it's not surprising that it's related to all of these outcomes, ranging from mental health to very physical outcomes. So she's really looking at it as almost a universal marker of health. And then personally, when I first came to the lab, I had my uric acid measured in saliva. I was immediately postpartum, very stressful time, and I was new faculties trying to set up my lab, and my uric acid was through the roof. And I was just so shocked, and it really helped me prioritize my health. And then 6 months later, took care of an ear infection, all of these things that a new mom doesn't do for herself, and I prioritized it, and 6 months later, my uric acid was way down. So the ability to bring this into physicians' labs to help monitor people at home and then have more individualized kind of tools for medical treatment and individualized treatment is what we're really passionate about at ISBR. We have a table with our fantastic postdocs there. Please go see them and get a stress ball with the saliva, and I'm going to stop there. All right, thank you so much.
Thank you so much, Kristina. Okay, so next up is Shahrdad Lotfipour, and he is coming to us from emergency medicine, and we'll be talking about the new UCI Behavioral Neuroscience Testing Core.
Well, thank you so much for the invitation. It's an honor to be here speaking in front of all of you guys today. So I'm going to be talking about the UCI Behavioral Neuroscience Core. It's so new that it's actually currently under construction. But if you wanted a little bit more information about it, we've developed a website for it. It's www.ucibtc.org. And we have initially started a Twitter handle, so happy to have you guys involved and learn more about it. I'm going to be the director of the UCI Behavioral Core. I'm also the director of the Translational Addiction Laboratory here at UCI. So just a little bit of a background on it. One thing to highlight is that collaboration is at the heart of our story. And in wanting to develop a behavioral neuroscience core at UCI, there's two things that you absolutely need. You need space and you need equipment. So fortunately for us, we've been able to acquire about $1 million worth of behavioral equipment through the assistance of myself and Dr. Mike Yasa, who are both fellow and director of CNLM. And we've capitalized on a major opportunity to work with Dr. Jen Precher, who had been initially allocated space and funding support for instrumentation of a new optical imaging core. And this was part of 600 square feet of the UCI Biological 3 building. It's part of the shell space, and I'm going to show you a photo of that. And it's 1/3 of the 1,800 square feet of available space that the UCI BTC is going to be part of. And we've, by acquiring the additional equipment for the behavioral core, it provided the incentive to renovate the entire shell space and develop the UCI BTC and the new optical imaging core. And it's not really so small because we've been actually able to get a major cap renovation grant in the amount of $1.667 million to develop this facility for the UCI Behavioral Core. So we're really excited about that. And of course, to be able to do so, you need a lot of support from a lot of different individuals, all of which are listed here for your review. So the UCI Behavioral Neuroscience Core, there's a major need for this. We need a state-of-the-art, high-throughput facility with video monitoring and recording for rigorously testing rodent behavior. This includes cognition, like learning, memory, and attention. We can measure affective measures like emotional, social, motivational functions, and we can evaluate sensory motor functions. And as part of this, we need dedicated expertise in small animal behavioral analysis to be able to provide consistency across a number of different testing modalities. So there's great need for the behavioral testing core at UC Irvine, especially for recently hired faculty as well as new recruits. And without a behavioral core, what it inhibits is that it limits you, the investigators wishing to use the behavior in their research projects, because you need funding to be able to develop the behavioral core, and you need space to be able to set up the behavior. And you need to also be trained to use that behavior or eventually collaborate with someone to be able to use the behavioral core. So UCI has actually never had this type of facility in the past, which is actually uncommon in our R1 institutions. So this allows us to leverage the facility for faculty and student recruitment, enhancing our competition against our peers by providing access to such resources. And then hopefully that reduces the need for funding, dedicated space, training, and the need for collaboration required for through this behavioral testing core, which I think will lead to reductions in inefficiencies related to these various types of concerns. So this is pictorially for your review the type of equipment that we've acquired, and it's also on our website, and I'm happy to speak to you guys more specifically about the individual components that we have that are going to be in this facility once it's developed. And ultimately, as mentioned, we're going to be evaluating anxiety and depression, learning and memory, social behaviors, sensory motor tasks, and we have secured software licenses to be able to run these various types of services. And in the future, what we hope to do is perhaps integrate it with in vivo electrophysiology, in vivo optical imaging, in vivo optogenetics to be able to enhance and leverage the behavioral core even more. This is the location where the facility is going to be based. It's in the Bioscience 3 basement shell space. It's laboratory 510. It's about 1,800 square feet, and 600 square feet will be for the imaging core and the grant that we've been able to get through the major CAP grant to develop this facility through the help of the Vice Chancellor of Research and others. So I'll be the manager of this facility, and then what we aim to do in the future is to be able to develop to have a PhD-trained behavioral neuroscientist to help with the behavioral testing, as well as hopefully a behavioral technician, assistant manager that would assist with the behaviors. And we can, in theory, start to develop a recharge rate where we can either work with you specifically to give you access to the facility based on using the equipment, or we can do the behavior for you at a particular cost, which is modeled after the UCLA Behavioral Testing Core. And in the future, what we hope to do is be able to custom-made optogenetic feral implants, in vivo optogenetic integration, in vivo electrophysiology, in vivo optical imaging, and be able to think about a P30 grant very similar to the core grant that the Salk Institute, for example, has to be able to support the core system for behavioral testing. There's several people that are involved that have helped us get to this very moment that are listed here. And if you have any additional questions or comments, feel free to visit our website or see me afterwards. Thank you so much for your time.
Okay. So now, for the last speaker in this session, I am very happy to introduce Mr. Ken Beal. Ken has, and his family, as many of you know, have contributed greatly to UCI campus, in particular in nurturing I think a transformation in the relationship of UCI to the business community here in Orange County and beyond. And so he's going to be talking today about a new biotech startup incubator, university lab partners, and also about partnerships with UCI on developing entrepreneurial workforce programs for engaging students with the business community. I know I'm speaking for myself, and certainly many of my colleagues who are brimming with ideas for new ventures, that it's really a treat to have you here, Ken, and hear about where you've been and how you've gotten here. And also, we're very excited to hear about the vision you have for our community. So, why don't you come on up? Thank you so much, Ken. We can just advance here, and then there's the projector.
Great. Okay, great. Well, thank you, everybody. It's always dangerous to be the last person between you and lunch, so I'll try to make this on target. But and I'm really excited to participate in your Brain launch event and to see the analogy you've made to the moonshot, because the space program is very central to our family. My dad spent his career with Rockwell and got started, I think he started work in 1961 or something, and the Apollo program was in full steam, and then that transitioned into the space shuttle program, that transitioned into aerospace work with the government, making various aerospace systems, commercial and defense. And so why is that important? I mean, we have a real interest as a family in seeing Orange County reach its potential. And we are strongly of the belief that for Orange County to reach its potential, UCI needs to be at the center of that. My dad went to San Jose State. I'm a UCLA Bruin. My brother's a Cal Bear. So we're related to UCI tangentially, but we're just very focused on how can Orange County reach its potential. My background is in the real estate business, and 30 years ago, if you looked around the county, you'd say, gosh, Orange County is a real estate-driven economy. You looked at the Irvine Company and several other handful of developers that created a strong business in that. But today, when you look around, it's not that way at all. Largest public company in Orange County is Edwards Life Sciences. You look at Allergan, you look at these life science, medical device, and med tech companies that have become the real core of what the county is and what we believe the county is going to be in the future. So why did we get started with University Lab Partners, and how do I get my picture off of this? University Lab Partners was really the culmination of our effort to get involved with the campus and encourage innovation and entrepreneurship generally. Initially, when my dad retired from Rockwell, the company made a gift to the UCI School of Art to create the Beal Center for Art and Technology, which is a collaboration between the engineering school and the fine arts school. And it was a real nod to both my mom, who's very into arts, and my dad, who's an engineer at heart. And it was a touching to them, and it spurred involvement by them with the university philanthropically for many years that continues today. There was a gift that was made to the business school to create the Center for Innovation and Entrepreneurship there, which was now leading the charge to create the first one-year master's program in the UC system, a master's in innovation and entrepreneurship. So not an MBA, but it's taught in the graduate school, and it's this September was the first incoming class. But while we were excited about how some of those initial activities were progressing, the itch we were really trying to scratch is how can we help UCI, which is doing roughly $400 million a year in research, how can we help translate that research? How can we help commercialize that? Fully understanding that there's a lot of research that's basic research, and that's great, and the university system is the place for that. But there's also, in our minds, opportunities to commercialize or translate that research, and we thought one element that was key to that was to get the business community involved. The business community knows how to bring things to market, they know customer needs, they know how to build a business. And so even though we were very active at the Center of Innovation and Entrepreneurship at the business school is not really core to where the inventions are happening. They can be part of the team, but they're not the team. So we started to then look at how translation and commercialization happened at other schools around the country. We looked at Stanford, we looked at UCLA, we looked at Columbia, and that effort resulted in the creation of UCI Applied Innovation. So I know the Vice Chancellor or Vice Chancellor of Research, Pramod, his predecessor was very involved in helping us look at these other schools. And what came out of that study was that rather than having the Office of Technology Alliances, I think was what it was previously known as, be buried kind of in the bowels of the Vice Chancellor of Research's responsibilities, how do we elevate that? How do we make it into an entity that is not only serving the faculty and researchers and students on campus as customers, but also connecting with the business community in a way that makes it easier for them to understand what resources are on campus and vice versa. And so that led to the creation of Applied Innovation, as I mentioned, which was stood up about 5 years ago. And it took we made an office in UCI Research Park, just up California, near the intersection of California and Bison. And it was there for an important reason. Rather than putting it in the center of campus or somewhere in an academic building, the idea was to put it off campus. UCI Research Park is technically off campus, although it's just across the street, and put it in a position where the business community can access it easily. I mean, back when they designed UCI into a circle, kind of inward looking, some would say, the 73 freeway didn't exist, the airport wasn't as big as it is, but now if you look at UCI from a business person's perspective, the front door of the university really is Bison coming right through UCI Research Park. So that was where they put the first home for Applied Innovation, and that was 5 or 6 years ago. Richard Sudek is the executive director of Applied Innovation, and he manages all the intellectual property of the university. So when you go to him for contracts, industry-sponsored research contracts, conflict of interest issues, any of those issues, they are managed from his office. And Applied Innovation grew and was really actively supported by the chancellor and the provost, and it was exciting to see it grow and get more involved. And it was also put at that location so it could serve the community as well and its constituents, as well as the university and all of its customers. Because at the core of what we believe, again, is that if the business community can get involved with what the university is doing in a way that's mutually beneficial to both, then 1 + 1 can equal 3. And so we started to support back in about that time, we started to support some faculty members at the School of Physical Sciences, Information Computer Science, with just non-dilutive gifts, grants, saying, we sat down with Dean Jan in the School of Physical Sciences first, and we said, we've been supporting you for years in a way because my dad is such a, lover of the physical sciences. And I asked my dad, what are they doing with the money that you're giving them? And he said, oh, I don't know. And I go, well, why are you giving them the money? What do you want them to do? And he goes, well, I like to go to those breakfast series they have. And I said, Dad, you can go to the breakfast series. I go, what would you like them to do with it? And he goes, well, I'd like to encourage faculty members or researchers who are doing something that has some potential for commercialization. And so 6 or 7 years ago, we went to Dean Janda, who was at the School of Physical Science. We said, we're going to give you $60,000. Go out to your faculty members with an RFP, see what comes back. Well, 3 people put in an application for that. And we said, well, okay, we don't want to make a decision. Let's split it 3 ways. And one of the faculty members who received the grant was Greg Weiss from the School of Chemistry, or Department of Chemistry. And lo and behold, 4 or 5 years later, he started a company based on that research, a little offshoot of that research, which he said at the time was really important because sometimes research grants are written so specifically that you can't do some things that might be on the cusp, on the edge. And he used that to do a little bit more research. And out came a company, and he called me. I'm in the real estate business. He said, well, Ken, I've got a problem. I got to knock on the door, and the Conflict of Interest Committee has visited me, and they say they want to know more what I'm doing with this device over here, and how is that related to the research? And I've told them, well, it's a company that I'm trying to start. And they said, well, that's great, but you can't do it in your academic lab. And so he called me and said, well, how can, is there a lab that I can find some space in? You're in the real estate business, can you find it? And I said, I'm sure I can. So I looked around and I realized you can't in Orange County. I mean, if you want to get small lab space, you're going to go to San Diego or you're going to go up to LA. And I thought, that doesn't make any sense. And so we started to look and research what was out there. We went to J Labs down in San Diego. We went to Bio Labs in LA. Well, now they're in LA, but they were also in San Diego. And we studied them. We tried to get other people to do it. We worked with some of the, Suzanne Sandemeyer at the School of Medicine was very helpful. And I said, just give me your worst building on campus, and I'll just give me a corner of it, like in the basement, 600 feet. Give me something started. And there was always a reason you couldn't do it. And so to fast forward to a year ago, applied innovation was growing. Industry-sponsored research has grown in the last 3 or 4 years from $12 million a year to $25 million a year. And I don't know why it can't grow to $100 million a year. And so that, I think, energized the administration to say, hey, we need to support this more. Broadcom, who was the tenant in 1 million square feet of space at the end of California going up the hill, vacated, and all of a sudden the Irvine company who owned those buildings had them all back. They were all vacant, and Applied Innovation went to the school and said, we think we can do more for the community and more for the school if you can help. They were now in 2 different spaces. Let us come together and consolidate our space and expand it, and we think we can do more. They got approval to do that. Richard Sudek, the director, said, we also need a wet lab incubator because we've got all these projects, and faculty members need a place where they can be close to their academic activities but still run over and do something that has some potential for translation. And the school said, we just can't do that. There's all these different demands you can imagine, so it didn't get funded. At the end of the day, we decided we were going to fund it. We thought it was important. And so we have leased half of the 3rd floor. So Applied Innovation is in a new building that they're just moved into this week at 5270 California up the road. And they leased the UCI has basically leased the whole building except for our 17,000 feet. So we took a big bite, signed a 10-year lease, and decided to try to develop a co-working space with wet labs. And so I think I might have a little bit of a video. We've covered that. I'll come back to this. And it's important, I guess, to note too, is the Wet Lab Incubator, University Lab Partners is our name. We're independent from the university. We're a nonprofit. We're an activity of a foundation. And we're one of the only ones we know of, frankly, around here. So our goal is to help accelerate commercialization activities. And also, as we got into it, we realized there's a great opportunity to create workforce development type activities, apprenticeship type activities. We met Sunil through his company Translucence, and we learned more about the NeuroScholars programs, which maybe some of you are involved in, and we thought, gosh, that's interesting. Our business model is in a co-working, we've got 66 individual lab benches, we've got 11 private lab suites that are about 200 square feet, we've got 17 private offices, and then we've got 21 cubicle spaces. So we're pretty flexible, and our business plan assumes we never get fully occupied because, again, we're not here to lease space to Translucence for the next 5 years. We're hoping they come for a year, they work on their science, they work on their business proposition, they work on financing and finding partners, and then they move on and they make room for the next person. Or, it doesn't move on, and we tell Sunil, it's been great, we love you, but you need to go back to your academic lab now. And so we're always trying, but we realized we've got space here, we've got equipment, and we're worried about Orange County. I mean, we have a low unemployment here in Orange County, but if you look at the types of jobs that have been created over the last 10 years, they're not the kind of jobs that you want to create. There's nothing wrong with hospitality jobs and leisure jobs and food service jobs, but they don't pay enough, frankly, for somebody to stay and live here. And so what if we could do some workforce training and make connections to the business community? Because if you asked Edwards Life Science or you asked Massimo, who's making devices or diagnostic tools, what's your biggest challenge? Well, it's people. And so anyway, that turned out to be not our first strategy, but evolved into our second, is that we can work with people who have existing programs like NeuroScholars and figure out how we can complement those and maybe add on to them. I'd like to show you a little video. I think it is... Here we go. I don't know how to start it. Maybe that'll go. Oh, there we go. Well, that's the floor plan. But let me see if I keep going here. And so again, you can see there's 3 bays. So what we've done, we didn't know who our tenants were going to be. We didn't know if they were going to be more biological oriented or if they were going to be medical device oriented. The first tenant we signed up was the previous chief scientific officer at Edwards Life Science, the developer of the first transcatheter heart valve that Edwards ended up buying his company 15 or 20 years ago. It's now $1 billion business. He's left Edwards, and he's taken 4 engineers with him, and he's leased some space there. So we made the right side of the lab. We think it's more fabrication. We've got laser welders. We've got fluid bio. We've got stuff a real estate guy doesn't know what it is, other than it's expensive. And so we've kind of said that's going to be more of our fabrication side. In the middle, it can go either way. We're not sure. On the left, we've got a fume hood bay. We've got a microscopy room. We've got a freezer farm. We've got tissue culture rooms. And so we think, biological more on that side. We'll see what happens. We've been really excited. Chris Hughes from the School of Biological Sciences, Abe Lee from the School of Biomedical Engineering have a company that's developing tumors on a chip and vascularizing them and doing drug discovery work with them. They lease L6 and L7 at the top. Those are our private lab suites. All this down here in purple, that's just in a general office area. So think we work without all the drama and just co-working space. So we're set up we're trying to take the friction out of commercializing. We're also trying, and the reason I'm so excited to get invited here today is UCI to somebody on the outside is very opaque. I mean, just sitting here and listening to these presentations, it's just intimidating for non-scientists. Now, but we know as the community knows there's an amazing resources on campus, but how do you access them? How do you, and I can tell you from firsthand experience, to get a recharge agreement signed, it can be complicated because that one is different than that one is different from that one. One of them talks about IP, the others don't. I mean, it is not like getting on your iPhone and saying, I agree, and off you go, and you just pay your $70. So we're here, Karen Cook is here in the audience somewhere. She's our ecosystem director and really is the the boots on the ground as we start, and we're going to open in December, by the way. But we're trying to learn more about the different facilities on campus so that we can be that clearinghouse. When one of our tenants says, gosh, did you know anybody who can run a mouse model on a brain with a lightscape like that first? Then we're going to say, yes, we know where to go. In fact, we know who to call. This is the agreement that they've already signed 100 times. Your insurance requirement that we made you sign for us is totally it's aligned with theirs. So we're trying to take the friction out of that. We're trying to be, I say sometimes we want to be the concierge at the hotel. When you walk down in a big city you're not familiar with, and you say, where's the best place to have Chinese food? And they've got 3 options. And so if one of our residents says, do you know anybody who could help me with this? We may not know exactly what this is, but we're going to find it and make it easy. And so that's our goal. We're excited. We're doing this to really honestly further the mission of UCI Applied Innovation. They are, a group of people that are dedicated to working with the business community and with all you faculty and researchers on campus to try to help make connections to not only just help you further along your research, but if there is the potential for commercialization to do that as well. So
let's see, maybe the video can play. I think just one more button press.
There we go. So again, we're just in one of the old Broadcom headquarters, just right up the road. This is taken a couple weeks ago. We're further along. We're going to be open on December 20th. So from a tenant standpoint, you've got conference rooms, reception areas, pantries for food, centralized Wi-Fi, printing, all that stuff. This is one of the bigger bays that'll have we have 66 standard 6-foot lab benches, snorkels and compressed air and different things, water. We spent $1.2 million on equipment, so we've got probably not exactly the right things because we don't know who our tenants are, but we've done our best. We're applying for these are some of the facilities just outside, which is if you haven't been over there, it's really nice. There's a new restaurant that just opened up. All this is available for tenants. So if you join and you become a resident of University Lab Partners, you get a free gym membership too. So that'll help the creative juices. There it is. And so it's really been great. The Irvine Company is a for-profit entity, obviously. We signed a 10-year lease. I can tell you they've been extremely supportive of what we're doing because I told them, I said, if you just stops here, then all we've done is we've pushed this problem a little further down the road. What we're hoping for is that we can prove out the model that there is demand from innovators and entrepreneurs, and we can grow companies and grow jobs and grow value for our community and hopefully drive more real estate needs for the Irvine Company. I said, you need to step up and build a building that has 1,500 to 2,000 square foot individual labs so that Sunil's successful company can grow and move down the road and we can bring in the next group. But anyway, I'm happy to answer any questions if there are any.